Let's get real. Let's get loud. On America Out Loud Talk News, this is the McCullough Report and Focal Points Substack. It's a great pleasure to invite to the program Dr. Wafik El-Deiry. He is a cancer specialist, and we're going to talk about his findings in a recent paper published in OncoTarget and broadly about the whole issue of cancer trends and what we're seeing post-pandemic. Dr. El-Deiry, welcome to the program.
Thank you, Dr. McCullough. It's a pleasure to join you this afternoon. So how would you like to do this?
Well, why don't you tell our audience just a little bit about your background, what you do on a day-to-day basis?
Yeah, so I am a practicing physician scientist who wears a few hats. I am the director of the Cancer Center at Brown University. I'm an associate dean at the medical school. I chair a global consortium in precision oncology called the WIN Consortium. There are two things about me and my career. There's a gene named after me, WAF1, discovered in the early 90s when I was at Johns Hopkins. Nobody cared what you called a gene in the early 90s. This one turned out to be important. It's how cells stop dividing when stressed or treated with radiation or chemotherapy. The other thing that I focused on for most of my independent academic career has been the innate immune system and its connections to tumor suppressor genes. So in the mid-90s, my lab discovered trail death receptor 5 as a p53 target. And this was my entry point into the innate immune system and how we may be able to harness it for cancer therapy. And eventually, we screened for molecules that can stimulate this trail pathway. And I'm happy to say that a drug we discovered in the lab 18 years ago, the same molecule was just approved by the FDA last August as the first ever treatment for aggressive brain tumors with H3K27M mutations, these middle of the brain cancers that respond poorly really to all available therapies, not a place where it's easy to operate, and essentially they're treated with radiation and experimental therapy at this time. And so, I spend a lot of my time trying to come up with new cancer therapeutics because, I didn't wake up one day saying, let me discover a drug to treat brain cancer in humans. It just turned out that way. Why hasn't it worked in other tumor types? How can we get it to work better? What more can we do? So I'm involved, in basic research that is translational with a goal of bringing new treatments to patients. And that's what I do.
That's an incredible life, incredible story. I don't think I've ever talked to anybody who has a gene named after them.
Yeah, for years I thought this is going to be the most important thing I would ever do, which is kind of depressing, as a junior faculty member years ago. But fortunately, having impact on patients is incredibly meaningful and very fulfilling. And so, I enjoy what I do. And, when this COVID pandemic started, I had certain expertise that I wanted to apply to specific problems we were facing. So back in 2020, we had this rip-roaring pandemic, and we knew a little bit about how the virus infects cells through ACE2. And so I immediately started to be interested. Can we block that somehow? Can we reduce infectivity? What can we learn from what we know about the innate immune system in terms of cytokines and disease severity? And then, all along as a cancer specialist and expert in cancer biology, we all know viruses can cause cancer. It's not too much of a stretch. Is there anything going on with the COVID virus that might ultimately lead to tumors developing? What do I know about that? Well, I've studied P53 for 30 years. I've discovered genes it regulates. And we know that viruses, well-known virus, human papillomavirus, other viruses target tumor suppressors like P53 and RB to transform cells and eventually lead to cancer. Can we look into that? And so that has been the type of research that I was involved with. So early on, back in 2020, by the summer of 2020, we published a paper because we discovered that a class of drugs used to treat cancer, MEK inhibitors, um, suppress ACE2 expression. And so this is a way to attenuate infectivity. And by the way, in that same paper, we observed that remdesivir increases ACE2 expression. And, that might be something worth knowing about. Some of our other work here in Rhode Island, we accessed blood samples from patients in the emergency room at Rhode Island Hospital through an approved protocol and did cytokine profiling and some algorithm to make sense of it. And I learned about something called macrophage activation syndrome, which I'd never heard of. But this cytokine profile correlated with disease severity and gave some insights. And then we eventually got around to doing some of the P53 experiments, which we published in, I think, April 2024. We looked at what happens to P53 function if we introduce spike into cells. And we were able to show using reporter genes or endogenous P53 targets that spike attenuates P53 transcriptional activity against reporter genes or its endogenous target genes in response to DNA damaging chemotherapeutic drugs. We showed that overexpression of spike makes tumor cells less sensitive to chemotherapeutic agents such as cisplatin or 5-fluorouracil. And we started to get some very early indications about the DNA damage response or the recognition of DNA damage, but really very, very early. All of that data was there, suggesting that more needs to be done to look at this.
Mm-hmm.
And then by the summer of 2025, I was invited to serve on the COVID immunization working group that reports to the ACIP committee. The working group is headed by Retsev Levy and has a group of experts. I think I was invited due to the cancer expertise, as well as some insight into clinical work, clinical trials, informed consent, and so on. And so during the summer and fall, I dug into the literature related to what do we know about COVID infection, COVID vaccination, and cancer. And, I worked with colleagues on the committee, and then Dr. Cooper Wasser at Tufts and I decided to write a review about what we had found and what we had reported to the ACIP working group, as well as the ACIP meeting in September. And what we said to the ACIP is out there. The video is there of what we reported. And essentially what we were reporting is that with the COVID vaccines, the mRNA vaccines, there were some unexpected observations having to do with issues of biodistribution, issues of impurities, issues of certain mechanisms with the pseudouridine modified mRNA as far as ribosomal frame shifting, immune effects with IgG4 class switching after 4 doses of COVID mRNA vaccines, and uncovered case reports, case series, some population studies around cancers that have occurred in patients with either COVID infection or COVID vaccination. And I would say, if you look at what we have found, there have been a number of reports of cancers that have occurred in temporal association with COVID mRNA vaccines within weeks to months after these doses. But I would say it's more than a temporal association. There's sort of a spatial association as far as cases of a case of sarcoma that occurred near the injection site after a second injection. I noticed that there is apparently, in what's reported, some enrichment or more than would be expected as far as lymphomas. There were lymphomas reported and more than I would have expected, and some involvement of regional lymph nodes, some skin cancers, some breast cancers, head and neck cancers, glioblastoma. There is a case series of patients with pancreatic cancers, and when you look at the IgG4 levels, the higher IgG4 levels that have been attributed to class switching associate with worse prognosis in those patients. There have been some population studies. I'm going to talk about look at the population study in Korea, and I think one in Italy. I'm not going to say much about those. Those have been critiqued by people on social media and other platforms. And I think, importantly, we haven't had a reliable study on U.S. data of cancer outcomes since the publication, or, I would say, I guess it was published in December. There's a study out of France that I think was published in one of the JAMA journals, maybe JAMA itself. I'm not sure right now. But anyways, it was millions and millions of people in France who were followed for at least a year. And that study didn't report a particular signal in cancers. And I thought at the time, well, that may be reassuring, but I've also learned from colleagues that there were some issues with that paper as far as potential biases. So, for example, I think vaccination was associated with reduction in accidents or suicides and things like that you wouldn't necessarily expect. Interestingly, after publishing our paper, I was contacted directly by a member of the Japanese government who is a representative by the name of Kazuhiro Haraguchi, who was also a minister and has been in government for years, that he personally developed a diffuse large B-cell lymphoma 3 years ago, and his lymphoma metastasized to his tonsils, and the metastases were biopsied and showed expression of spike protein and absence of nuclear capsid protein, consistent with a post-vaccination phenomenon. I personally would not expect to see spike protein expression in any tumor. In the paper we published, there was a case of breast cancer that metastasized to the skin, where a similar phenomenon was shown of spike protein expression and no nucleic capsid protein. In the end, both in reporting to the ACIP working group and ACIP and in this paper, there are gaps in our knowledge. There are things we need to study more. And I think one of those involves forensic evidence, pathology evidence, more looking at these tumors to try and, one of the issues that comes up has to do with causality. Our paper does not address that. And I, in a way, come at some of this evidence from the point of view of a clinician with some clinical trial experience, where in the conduct of clinical trials, when there are adverse events, those are reported, consent forms are updated, and you don't need causality. You just need reasonable suspicion that things may be related. And what's available is that temporal and spatial association. I mean, some would say in large populations where billions of doses have been given, these are potentially random events that occurred, but they did happen and they should be known. And ultimately, my view is perhaps these occurrences are rare, but I know from what I know that as far as cancer and other diseases, there's a great deal of heterogeneity in the population. So we're all different from each other. The chance that we will develop cancer varies. It's affected by our genetics, our environment. And so I think that there may be individuals at higher risk from adverse outcomes, potentially including cancer. I also became aware, Dr. Patrick Soon-Shiong has spoken publicly about seeing cases of cancer in younger individuals, early teens or even less than 10 years old, pancreatic cancer, colon cancer. I recently watched a documentary interview he was giving, where he invited some patients with head and neck cancer who had HPV. And I remember one patient who had HPV head and neck cancer 8 years earlier and was in remission and got a COVID vaccine. And apparently his head and neck cancer relapsed with liver metastases, which is interesting and important and deserves some thinking about why did this happen, what more can we learn. But for me, it sort of feeds into viral mechanisms by which cancers can develop and whether there's cooperativity, for example, between HPV and COVID or COVID spike protein in disease pathogenesis. I mean, we really have to start somewhere to describe what is being seen and then come up with scientific directions to unravel mechanisms. But I also think we need to fill in gaps in our knowledge with what is happening in individuals who have developed cancers. I think we really need much more of a database where this temporal and spatial association is there, but also the pathological analysis of these tumors as far as spike and nucleocapsid and other mechanisms. I think causality is very hard to get at. It can take years. But we are where we are. I mean, it's also 5 years out from the pandemic. There isn't a rip-roaring pandemic with high mortality rates every day. The mRNA vaccines have just been rolled out to the general population for the first time without long-term follow-up studies. And so, 5 years later, I think there are some nuances. Also, if you look at this French study that just came out with millions and millions of people without a cancer signal, the vast majority of the people who were in that population study had 3 or fewer vaccine doses. So very, very few. I mean, way, way less than 1%, much less than 1% had 4 doses. So, some of these nuances are important. It's really time to have some of these nuances fleshed out a little bit better in terms of who is at risk and what are the clinical situations where risk is increased based on the host. And of course, there have been other things that are unexpected that have been reported, such as DNA impurities, plasmid impurities, which could have additional consequences. I think these are things that could be addressed. I can say that when we published our Spike P53 paper, there are 2 types of oncologists, those who are optimistic and those who are pessimistic. I tend to be optimistic. And so in writing the discussion of that paper, I tried to come up with what could we learn from a situation where spike protein as part of a vaccine suppresses P53 activity. One thing we could learn is if we want to design and develop safer vaccines, we should try to get those vaccines to optimal antiviral immunity, but limit collateral damage to host defenses, including ways we protect ourselves from cancer. That's not such a crazy idea. I think it's a lesson learned. If I was developing a spike antigen for a vaccine, I'd want to test it in some ways to just make sure that it's not doing other things that we may wish to avoid. And so, that's one thing that came.
Well, let me ask you. Messenger RNA has been in development for many, many years. There's a paper by Lalani and colleagues in British Medical Journal, chronicling the timeline back from the 1980s. And, government agencies have been involved. One of the military agencies, DARPA, announced a program in 2012. It's called the Adept P3 program. It's still on their website and today. And it says, we're developing messenger RNA vaccines to end pandemics in 60 days. So my question is, so obviously this idea has been around for a long time, clearly in government programmatic language testing. Have you seen convincing preclinical oncogenicity data regarding messenger RNA products?
I haven't seen such data, but I think it's important to be clear about the mRNA. The pseudouridine modified stabilized mRNA isn't your average mRNA. I mean, mRNA, you breathe on it and it degrades easily. And that's clearly an issue. So, the Nobel Prize winning discovery was pseudouridine modification stabilizes the mRNA. And so this has enabled the incorporation into vaccines. And I think if you look in the literature, I was at some government website recently looking for the history of use of mRNA vaccines, and I can't find any prior human use of pseudouridine modified mRNA as a vaccine strategy. I really think it was tested first in humans in the setting of this COVID pandemic. So I'm not seeing that. I would say that.
Well, hang on, before you leave there, I'd make the case that we should have oncogenicity testing. Moderna has a messenger RNA RSV vaccine that's in widespread use now. I don't think that's undergone any preclinical testing for, can it be related to cancers? And that's not an emergency. We have a vaccine, and we've got other ways to treat the illness.
One of the things that I found in my investigation of what do we know about these mRNA vaccines and COVID and cancer, at some point I went to the FDA's website and looked at the package inserts of all the approved mRNA vaccines. And, they all use very similar language about lack of information about fertility, about lack of carcinogenicity studies. And I haven't checked in the last 48 hours, but they say so up to the present time. Okay. 5 years later, the package inserts, that's what they say. So, and I think we're all aware that the longer term human data up until, these larger population studies from other countries have all been limited up to like 42 days of outcomes data related to safety. One of the things that's coming out of the observations and our paper and other information is if we're going to pursue mRNA vaccines, including in cancer, by the way, I mean, more things need to be done. And I would say, I'm sure you're aware of the Nature paper that came out in November saying that the mRNA vaccines may actually be a good thing if you have cancer because they can make you respond better to cancer immunotherapy. And the paper used patients with melanoma and non-small cell lung cancer, two types of cancer that already respond pretty well to immunotherapy. They're hot tumors, they have an immune response. And but interestingly, and what wasn't stressed or really discussed very much is that the COVID mRNA vaccines that were used in these patients increase PD-L1 expression as a primary effect. Now, PD-L1 is an immune evasion mechanism, and that is tumor promoting as an effect on its own, absent anything else. And then you come in with immune checkpoint blockade, and you are able to demonstrate results. Now, the COVID mRNA vaccines in that paper and any other paper have never been shown to have a primary anti-tumor effect. And if anything, the Nature paper that says they may be good shows increased PD-L1 expression. I pointed that out on social media. I learn a lot from social media by discussing some things. And it was pointed back to me that, radiation and cisplatin that we use to treat cancer also increase PD-L1. And hey, they're combined with immunotherapy as well. And so what's the problem? Well, the problem is radiation and cisplatin have been used for decades. They have primary anti-cancer activity. They're anti-cancer agents. We've been using them to treat cancer long before we even knew about PD-L1. So they're anti-cancer drugs that, by the way, they do this and we could take advantage of it. The COVID mRNA vaccines are not themselves anti-cancer agents. Moreover, the effect of the increased PD-L1 depended on type 1 interferon effects. And, if you know about melanoma, you know that interferon is an approved therapy for melanoma. So is this better? I mean, is this approach of being COVID vaccinated in some temporal relationship to when you might get immune checkpoint blockade, is that better than just being treated with interferon? It may be. My understanding is the plan now, as described recently in the cancer letter, is to conduct a large study in lung cancer, focus on lung cancer to test this further. And, my view on this is that in the context of clinical research, that's absolutely fine, as long as the consent form is clear about what we know, and as long as the protocol that's reviewed by the IRB is also clear about what is known to the present time. And if patients would like to participate, that's absolutely fine. I do want to add a comment that because I've, been accused incorrectly of being anti-vaxxer. I'm not an anti-vaxxer. I'm somebody who was very happy at the end of 2020 to receive the first vaccine dose. Back then, it was great that it happened and was available. There's no intent to discourage anyone. It's for me, it's all about informed consent. I think the public and our colleagues and everybody else can process information about risk and benefit. And I think what has happened should be known. It should be in consent forms. I mean, the package insert, by the way, says there's a risk of death from these vaccines. So, if you're okay with saying there's a risk of death, maybe you should be okay with saying, hey, some people developed cancer. We don't know how common that is, or some may be more susceptible to getting tumors. And, you should be aware of that. But if you and your doctor think that there is an upside and a benefit that outweighs whatever risks, then by all means, people should be able to get their vaccines, and they, shouldn't have to pay for it either. They should be covered. So I think that should be out there and very clear, because I think, for me, I'm interested in science. I'm interested in the truth more than anything else. And I think It shouldn't, this shouldn't be so political. It should be about what is the truth. I mean, the truth isn't going to change. It's going to be the same 50 years from now. Whatever is being observed, whatever is happening, the science is not going to change. And so, we should seek the truth and use what we know when we know it. And I think there's a lot of work to be done in this field. Vaccines, there are many options. I mean, consent forms should be clear about the availability of peptide vaccines, for example, which may have less toxicity. Although, when I was looking into Novavax, I noticed that the myocarditis is still an issue there. So it's likely, in my mind, this is personal analysis, that the myocarditis may be more, autoimmune than spike related, specifically in the heart. But then again, I don't know enough about these mechanisms, or there might be multiple mechanisms for all.
Well, I can tell you as a cardiologist, I'm dealing with myocarditis on a daily basis, and have been, now for 5 years. But we're going to take a break right here for our commercial sponsors, and then we're going to come back on the back side of this. And I want to ask you some specific questions. We've been talking to Dr. Wafik El-Deiry. He's a hematologist, oncologist, and really a wide-ranging discussion on cancer and oncogenesis, and really terrific content to, I think, fairly evaluate. He's laid it out there very clearly. You're listening to the McCullough Report. Let's get real. Let's get loud on America Out Loud Talk Radio. This is the McCullough Report and Focal Point Substack. We'll pause right here. Let's get real. Let's get loud on America Out Loud Talk News. This is the McCullough Report and Focal Point Substack. Dr. El-Deiry, it's rare that I get to talk to somebody with an H index of 135. Now, I'm just ahead of you at 137, so it's rare you ever talk to somebody ahead of you. And so,
all I can tell you is there's probably going to be a bolus of papers. I mean, it takes a while, but we keep trying to.
Oh gosh, I already have 100. I have 100 on the pandemic. I was the first to publish how to treat it. In McCullough protocol became the most widely used early treatment protocol, adopted by major medical society, and it went on from there and focus. And I've published now the most on the myocarditis. So I can tell you what's gotten my attention is the messenger RNA from the vaccine in the heart muscle tissue.
Congratulations on your
Okay.
It's not a small accomplishment. I mean, when I exceeded 100, I realized at that point very few people in the world get to do that. And so I think that is something that should be recognized. And there are a lot of people out there who want to diminish the impact or what this means, or it's just a number or whatever, but they're not truly our peers.
No, there's not. And certainly, our peers, many of them don't want to talk about this. We haven't seen grand round series on these topics, or important, CME series on, how to deal with these emerging issues. But what's caught my attention is messenger RNA in the heart for sure, and then spike protein in the heart for sure. And of course, inflammation, because the spike protein is there. Not only just in fluids, but it's expressed on cell surfaces. It's found in the cytosol. We have a paper in preparation right now, but it's widely known, where we have someone who did not have the infection. So he's negative against the nucleocapsid, and he has measurable Pfizer messenger RNA in his bloodstream 3.2 years afterwards. We use a reverse qPCR method to nail it down, and we have enough primers on it to know it's the vaccine. And then the full-length vaccine spike protein, which is held open in the prefusion conformation in a trimer, and there's a proline insert. So there's actually a way to molecularly figure out if it is a Pfizer Moderna spike protein and not the natural protein. We see fragments of DNA coding for the messenger RNA. So they come off the plasmid in this manufacturing process. So fragments coding for fragments of RNA, coding for obviously a piece of the spike protein. And then we see fragments for the SV40, the promoter, the enhancer, the inhibitor, the antibiotic resistance genes. So we see quite a blend. We've done skin biopsies in the same person. We find everything in the skin as well. So it's out there in the tissues. We have a bladder cancer patient where we have identified at least a partial code in the genome of the bladder cancer cells in a young woman, her 30s, that matches a part of the code for the messenger RNA vaccine. And where I'm going with this is, we're 5 years into this vaccine campaign. From the manufacturers, no bona fide cancer investigation from a preclinical perspective. We've already seen another messenger RNA vaccine roll out for for RSV. We've seen a whole series of boosters. And again, I haven't seen any major conferences, Bethesda meetings, anything from the National Cancer Institute on this at all. Outside of you and a few others, I'm concerned essentially that a potential cancer signal is being swept under the rug.
So, my comment is there are the manufacturers. We also have the CDC and the FDA who capture data and oversee products. And I think that there's an ongoing process right now through these different committees. The ACIP committee is certainly being attacked in some ways, even as they are investigating these phenomenon. And I think that I would like to not believe that information is being swept under the rug, but there's clearly a narrative about vaccine safety and some I won't say prohibition of just any other points of view or even asking questions. I mean, I've grown up in academic medicine doing science, and this is just not how science is done. I mean, I think we ask, things are observed, we look at available evidence, and we ask questions, and then we pursue this. I personally think there's enough there of concern. And, maybe the concern at some level in terms of safety is not very high. I just don't know the answer. But for some things, they can be quantified eventually. I still think that the heterogeneity in the population, to me, suggests that some will be more susceptible than others. So who is more susceptible and what is their risk? Versus perhaps the general population. Maybe that is, if you look at the general population, and again, the studies that are out there have their limitations, but let's say there is a valid, reliable study that just doesn't see much at a population level, that still doesn't get at the individualized risk that people are taking. But, I still think we need better population studies. I mean, I'll tell you this. I wish there were more published reports. Sometimes when I hear of things being described on social media, it can come across as maybe this is the tip of the iceberg. But at the same time, if there are hundreds of cases that are out there in these different case reports and case series, there may be more. So I think we need more. We need, the questions have to be asked before they can be answered.
I've been an editor of major journals for decades now. I am right now. My estimate is about 1% is getting through. About 1%. And when I talk to my colleagues, who are at major academic medical centers, which produce the bulk of the peer-reviewed papers, they've told me, listen, I couldn't get my department chairman to ever sign off on a paper concerning a vaccine side effect or be approved by the research department. So they're not even getting out the door. When they get out the door and they submit, and I've had papers where I've had to submit to nearly 100 journals for a paper. I've never had to do that before in my life. I've had papers not only be desk rejected, but they were distributed to associate editors, and then associate editors refused to handle them. Never seen that happen before. Refused to handle a paper. So there's something about vaccine safety that is taboo. So this is a good activity for artificial intelligence. So I asked artificial intelligence, I said, of all the papers on the COVID-19 vaccines in the big journals, let's say New England Journal of Medicine, Lancet, JAMA, how many of them in some way conclude that the benefits outweigh the risks? And the number came back at 380 to 0. There wasn't a single paper that said, well, maybe the risks in this situation, if you were to dial that in for statins or for doxorubicin or for back surgery, there's always a balance. There's always some, and we try to work it out. But with these COVID vaccines, there is no balance.
And it's interesting because there's also no liability as an issue. I've been in the publishing world for about 30 years on editorial boards. I was founding editor-in-chief of Cancer Biology and Therapy. I'm a co-editor-in-chief of OncoTarget. And by the way, I was, despite what people might say or claim, I had no knowledge of the review process of our paper. I was not involved. I will tell you this. We do have a paper in press similar to our cancer paper that will document, I think, like 2 years of interactions with other journals, including rejection post-acceptance and failure to publish. This is hopefully coming out in the not too distant future. It was still peer reviewed at our journal and improved still. And hopefully my saying this does not encourage anybody out there to initiate any new cyber attacks on our journal, because we did go through that in December and January. We've gone through it in the past. This time it was particularly intense. And I will say, given this opportunity, anybody out there who has anything related to vaccines and cancer, feel free to send it to the journal OncoTarget. It will get a fair review with constructive criticism, because I think we need more data to be publicly available in a balanced way, so that the scientific community can have a better handle. And I will say that I am also a little bit troubled to hear in the news in the last few days that both Pfizer and Moderna are sort of pulling back support of vaccine development. I don't think that's the goal. I think we should all be thinking more constructively. If mRNA vaccines are really, important developments that ultimately bring hope to patients and can be done more safely and effectively, then we should pursue that, and it shouldn't be. I'm not happy to see that either. I think if they have promise and can help people, by all means, they should be developed, and people shouldn't rush to abandon them. If they ultimately can help people. So, that's my outlook there. But I should also say one of the things with our paper is that the reports came from all over the world. It wasn't a hot spot somewhere. They really came from all over the world. And I think when you talk about clinical case reports, I mean, I've been asked the question, were they all in peer-reviewed, reputable journals? Often case reports by their very nature do not end up in, most of your top journals are not publishing case reports to begin with. And I think those who are in the field have the ability to look at a case report and determine, is this credible? Is it, I mean, it's really hard if somebody's going to make stuff up, but we assume that our colleagues who are submitting these things are honest about what they write down and what they show. And I do think some of the evidence, while circumstantial, goes beyond just randomness. I think there's maybe something there that we should study more.
Well, it certainly raises a concern. I'd like to see the same concern start to come from the National Institutes of Health and the National Cancer Institutes. I have real concerns on cardiovascular safety. We're not seeing that either from the NHLBI. And I think we have a long way to go. This has been a great conversation. We're going to have to leave it here. We've been talking to Dr. Wafik El-Deiry, who's a hematologist oncologist. He's a leader in cancer research, and he is the editor of OncoTarget, which is a journal. I'm the editor of the International Journal of Cardiovascular Research and Innovation, formerly editor of two major journals. But our journals do welcome, and we'll give a fair review to papers on this topic. And you're right, decades in the future, it is going to be what it is. And despite all the current wrangling right now, I know a lot of people just simply don't want to hear this, but they should hear it because the information.
Yeah, one other thing I'll mention is, as a society with our regulatory oversight, et cetera, we need reliable systems that capture the type of data that we'd like to be looking at. And a lot of this has to do with attribution, causes of death. What you put in is what you end up looking at. And so there's some things to deal with, and I'm personally optimistic that we will see progress in those areas as well as more information continues to come out regarding the limitations that need to be addressed.
Well, I hope some fundamental things. So vaccinated or not vaccinated, that's a hard binary variable. I'm not vaccinated, you are. So that's a hard binary variable. Our CDC has those data. We have the SEER registry of all the cancer registries. We have the ability to merge data. I mean, these are things that honestly need to happen soon. This has gone on for a long time now. And
I will add, there's vaccinated and unvaccinated. There's also history of known COVID infection, history of no known COVID infection. And I do think as time goes by, there should be more interest and attention to the number of shots, the number of boosters. I think when you're getting out to 5, 6, 7, and people are asking questions, I think we need evidence and data that is more focused on that situation rather than the billions of doses that were given in a different setting, basically, with regard to what has been observed as outcomes.
Well, for sure, I can tell you clinically, I document in the medical history every dose of a COVID vaccine, what brand and in the time frame given. Also, the number of documented COVID infections and then strongly suspected. I'm using kind of an indirect measure of spike exposure, which is the quantitative antibody against spike. And
By the way, Dr. McCullough, I can't remember if at one point I asked you directly. I mean, it's 2026, and I don't believe there is a clinical test where any physician can order a spike level. And it seems to me that we should have that, and I honestly don't know why we don't.
We're doing it in research. It's a very simple ELISA test. We do it in the Emodia lab in Germany. There's a company in Michigan that's announced through press release just a few days ago that they will have a blood test. This is not hard to do. I did another, it's a good AI search. I did an AI search in 145 countries. There is no commercially available spike assay, although it's such a simple test to do. I'm encouraging for all samples, all biopsies and tumor samples to be stained for spike protein, to have reverse qPCR to test to see if the vaccine is physically there in the tumor. It's been described at least in one colon cancer so far. And we need a lot of work. I hope you can have influence at the NCI. I'll try to have influence at NHLBI, because we do need to light a fire. There's a lack of alarm at a high level right now. That a lot of people think this is water under the bridge. These products are still on the market. I think the utilization is minimal. We don't have reliable data, by the way, on who's taking a vaccine now, because the CDC relies on what's called the NIS, the National Immunization Survey, which is a random phone call to households. And people kind of give, if they answer, they give their intent on taking a vaccine. So they're no longer reporting the actual administration.
I think one of the issues, and this is going to have to be dealt with as well, part of the narrative is concerns about mRNA vaccines and their safety and potentially rare events are affecting people's views on vaccines in general. And I think, that's problematic. I mean, we know of diseases that have been eradicated with vaccines, but this is a new terrain, a new chapter in modified mRNA vaccines. And what we're talking about and the issues shouldn't really reflect so much on some of the classical vaccines that have had impact on public health. So, that is yet another challenge, for people to understand there are different kinds of vaccines. The issues we're talking about are not your average concerns with the flu vaccine. Not that there aren't issues with how well those vaccines work, but I do think that those distinctions get lost sometimes by people who just say, we're just doing a disservice to the community by raising these types of alarms, when they're really focused on a specific type of vaccine in a specific setting and different clinical scenarios and what makes sense and what's appropriate. And so, I think that also needs to be understood a little better.
Right. Completely agree. Dr. El-Deiry, we're going to have to sign off now. Thank you so much for being on the show.
Take care. Good talking to you.
Let's get real. Let's get loud on America Out Loud Talk News. This is McCullough Report and Focal Substack. I'm Dr. Peter McCullough.